Hello and welcome to Nikolay's genetics lessons, and here is a problem. A DNA from a strain of Bacillus subtilis with the genotype of tryptophan positive tyrosine positive is used to transform a recipient strain with the genotype of tryptophan tyrosine. The following numbers of transformed cells were recovered. Here is a genotype and here is a number of transformed cells. And here's a question: what do these results suggest about the linkage of the tryptophan and tyrosine genes? So imagine that here we have circular genome of bacteria, that is recipient, and here we have inserted fragment of the DNA of the other bacteria, and this fragment was inserted by bacteriophage. So bacteriophage wouldn't be able to insert the whole genome of the other bacteria because of the small size of its head, but would insert just a small fragment. And that fragment may contain 1, 2, 3, 4 genes. And in our case, we have a problem with 2 genes. So what might happen? For example, imagine that here we would have a locus for gene that is going to be tryptophan. So TRP positive here, and recipient would have TRP. And say somewhere here we would have another locus where we would have tyrosine, so tyrosine positive or tyrosine plus, how you like, that we can find on the fragment that has been inserted by bacteriophage, and in the genome of the recipient we would find tyrosine here. So what might happen? How this fragment may be integrated? We may have such situation, for example, when one crossing over would happen here, another would happen here. So what we are going to get? We are going to get this fragment integrated here, and this tryptophan plus would replace this tryptophan of the bacteria that is recipient. We also may have situation, for example, when we would have one crossing over here, another crossing over here. What going to happen in this case? This tryptophan would be still in the genome of the bacteria recipient, but this fragment would be replaced along with this gene. So we would have tryptophan and tyrosine positive in the genome of the recipient bacteria. What else might happen? We may have a situation when, for example, one crossing over would happen here and another crossing over would happen here. In this case, both tryptophan and tyrosine would be integrated into the genome of the bacteria, and we would have tryptophan positive here and tyrosine positive here. But let's take a look at different model. Once again, we have here the same genome of the bacteria. Here we have the same fragment that has been inserted by bacteriophage, and we have here also tryptophan, so TRP positive here, and tyrosine here, tyrosine positive here. And in the same locus we would have, of course, in the recipient DNA of the bacteria, tryptophan and tyrosine. So now what may happen here? Once again, one crossing over may happen here, another may happen here. And this fragment would be replaced, and both these genes would replace these 2 genes. What else may happen? We may have another crossing over here and here. Once again, these 2 genes would replace these two genes, and we would find this fragment to be integrated here. We may have different situation when, for example, we would have one crossing over here, another here. Once again, these two genes would be integrated together and would replace these two genes. And sometimes we may have crossing over here, say, and here. But because two genes are close together, such events when these two genes would be integrated separately would be greatly reduced if we would compare with this model where two genes separated by greater distance. Of course, there is more chances that these two genes would be integrated into the genome of the bacteria separately. So now we have understanding that the distance between two genes affect how frequently these two genes would be integrated into the genome of the recipient together or separately. If two genes would be close together, they would also tend to be inserted and integrated together. If distance would be greater, then we would have different variants when only one gene would be integrated. So now, when you have this understanding, let's return to our problem and let's take a look. For example, what we see here. We see that only one gene, so this situation, have been integrated into the genome of the bacteria. In the second example, we see that only tyrosine have been integrated into the genome of bacteria. And third example would be when two genes would be inserted together. So, can we tell that these two genes are close together or not? We can do it if we would convert these numbers into the frequencies, or if we would convert these numbers in percentages. So how we are going to do it? First of all, we have to add all these numbers. So 154 plus 312 plus 354. And we are going to get 820. Now we have to divide 354 by 820. And we are going to get 0.43. And if we need an answer in percentage form, we have to multiply this answer by 100, and we are going to get 43%. And this is going to be our answer. And actually, what this number tells us? This number tells us that these two genes are close together, and that's why these two genes tend to be inserted together. And we see smaller frequencies that these two genes would be inserted separately from each other. Why this information is important? It was actually important about 20-30 years ago when scientists were using this information in order to find gene linkage and also to build gene maps of the bacteria. But nowadays we can do sequencing of the genome of the bacteria in less than 1 day. So basically this example just demonstrates the linkage of the genes, that some genes can be located very close to each other, some genes can be separated by the greater distance, and if genes would be located next to each other, then such genes also would tend to be inherited together, or in the case of bacteria, if transformation would happen, also have a tendency to be integrated together. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates. And see you in the next video. Goodbye.